Hello guys, this is Dr. Possibility from Excel Academy. Please make sure that you subscribe to our YouTube channel and make sure that you register with Excel Academy. In this video, I'm just simply going to explain on the formation of the neurochome. So when we talk of the formation of the neurochome, we are just simply trying to discuss how the nervous system is being formed. We need to understand that the nervous system, it is made up of two types. We have the central nervous system, which is made up of the spinal cord and the brain. We also have the peripheral nervous system. But in this class, we are just going to focus on how the nervous system is created. Let's quickly get started. But before we get started, we need to borrow some knowledge from week 2 and week 3. We need to understand that during week 3, this is known as the gastration period. During the gastration period, we need to expect that 3 germ layers are going to be created. And the 3 germ layers are: 1, you have the ectoderm, which is coming from the epiblast. 2, you have the endoderm, which is coming from the from the hypoblast. And lastly, you have the mesoderm, which is coming from the cells of the of the ectoderm. With that being said, one thing we need to understand that the 3 germ layers which are created or formed during week 3, they are normally created by the process of gastration. And these 3 germ layers, they are normally originating from the 2 germ layers which were created in week 2. And the 2 germ layers are the epiblast and the hypoblast, of which, like I have stated, the epiblast will give rise to the ectoderm and the hypoblast will give rise to the endoderm. Then the cells of the ectoderm will start to divide, and the type of division which is normally taking place is the mitotic type of division. And this type of division, it is just leading to the increase in the number of cells. As a result, these cells will start increasing in number, and they are going to form the 3rd type of layer, which is known as the mesoderm. So this simply means that the mesoderm is the last germ layer to be created. So, like I have said, 1, the epiblast will normally become the ectoderm, and then the hypoblast will normally become the endoderm. When you talk of the endoderm is a very important germ layer which is going to give the lining of the GIT and also the respiratory tract. And most of these muscles of the GIT and also that of the respiratory tract, they normally come from the endoderm. Like I already said, the cells of the ectoderm will start to divide. And when these cells of the ectoderm divide, they are going to increase in number, leading to the formation of the 3rd type of layer, which is known as the intra-embryonic mesoderm. Remember, this is an intra-embryonic mesoderm. And this intra-embryonic mesoderm, it is a mesoderm or a layer which is between the ectoderm. So you have the ectoderm there and the endoderm. The layer which is existing between these two guys, it is a layer which we are calling the intra-embryonic mesoderm. The name has been specified because we have different types of mesoderm, which will be discussed in 3. Let's quickly look at this in terms of the diagram. So when you talk of this diagram, you need to understand that by week 4, there is already the formation of the morula. So this morula will normally divide into 2 layers. And these layers, you have 1, the outer cell mass. We have the outer cell mass, and the inner cell mass. The outer cell mass is known as the trophoblast. And then the inner cell mass is known as the embryoblast. It is the inner cell mass which forms the future embryo. And this inner cell mass, when you talk of week 2, it will simply divide into the epiblast and the hypoblast. When we talk of the outer cell mass, it is the one which is playing a role in the entrance or penetration of the functional endothelial layer of the uterus. And when you talk of this functional endothelial layer of the uterus, when these outer cell mass, which consists of the syncytioblast and cytotrophoblast, of which the syncytioblast forms the outermost of the two. And when these syncytioblast and cytotrophoblast are completely embedded into the endothelial layer, specifically the functional endothelial layers of the uterus, this will normally lead to the bleeding, which was normally experienced on day 28 on day 23 of the menstrual cycle. So let's quickly move on and look at this part. So this guy on day 6, we need to expect the blastocyst. The blastocyst, we need to understand that there is a fluid here which is filled up of some fluids coming from the inner cell mass, and this cavity is known as the blastocyst cavity. On day 10, which is week 2, so in week 2 we need to expect that there is the formation of the outer cell mass, which has to divide into the cytotrophoblast and the syncytioblast. Then there is a layer of cells. The first layer you need to consider is the hypoblast. So this is the hypoblast, which is normally made up of the simple cuboidal cells. Then we have the epiblast, which is made up of the simple columnar cells. The epiblast will normally divide, okay? The epiblast will normally divide. So when this epiblast continue dividing, it's going to create a cavity which is known as the amniotic cavity. So this amniotic cavity, it is formed within the epiblast, and that is during week 2. So with that being said, you need to understand that the name changes for these cells which are closer to the cytotrophoblast. So these cells which are closer to the cytotrophoblast are normally known as the amnioblast. Let me quickly write that. They are known as the amnioblast. So the amnioblasts are normally derived from the epiblast, and these amnioblasts are normally close to the cytotrophoblast, or you can talk about the trophoblast. During the third week, we need to understand that there is a division whereby this epiblast here, epiblast is going to become the ectoderm. The epiblast will normally become the ectoderm. And then the down part, which is having the simple columnar cells, which is the hypoblast, is going to change into the endoderm. Then the cells of the ectoderm will start dividing to cause a third layer of cell, and this third layer of cell is the one which is known as the mesoderm. So please make sure that you understand this and make sure that you are able to analyze these pictures. So you need to understand that when you talk of the ectoderm is a very important layer of the embryo, such that the ectoderm will give rise to the covering of the epidermis. Even the cells such as the melanocytes, So when you talk of cells such as the melanocyte, which forms part of the connective tissue, these cells normally come from the what? The nucleus. So when you talk of the nucleus is normally coming from the what? It's normally also being formed from the what? From the ectoderm. So please make sure that you understand that the ectoderm will also give rise to the what? To the nervous system. Now, this ectoderm will start making some changes with time, which we are going to discuss. On the other hand, when we talk of the endoderm gives the inner skin, which is just simply the what? The dermis. So the dermis normally comes from the what? From the endoderm. And this endoderm, it forms the lining of the GIT and also the respiratory system, just like I have already stated. Other thing that you need to understand that when you talk of the endoderm, you can also consider other organs such as the liver and the pancreas, including the what? The lungs, specifically the inner layer of the lungs. These organs, they normally come from the what? The endoderm. Then the mesoderm, which is the third part of which is the third layer of germ cells. And when you talk of the mesoderm, it's simply coming from the what? It's simply coming from the ectoderm. So when you talk of the mesoderm, it's giving the middle skin, which forms the skeletal system, connective system, and also the muscle tissue. Okay, let's quickly move on. This is just the diagram which I have already explained using the notes. So please pause the video, subscribe to our YouTube channel, and also go through this note. Like you can see, this diagram is a very important diagram you need to know. This diagram here, you can look at this part as the primitive node. So this is known as the primitive node. Okay, so this is the primitive node, and then this guy is the primitive streak, which you need to make sure that you know it. Then this layer, it is that the layer of the ectoderm. So this layer is the ectoderm from the what? From the epiblast, and then there you have cells of the what? Of the mesoderm. So cells of the mesoderm. And then lastly, you have the last layer, which is the endoderm, and this endoderm is simply coming from the what? From the hypoblast. So the hypoblast is giving rise to the what? To the endoderm, whereas the ectoderm is just simply coming from the what? From the epiblast. And the week during the week in which these guys are formed, this is just week what? Week 3. Okay, so let's quickly move on and look at the formation of the primitive streak. Remember, in this diagram I've talked about the primitive streak, and I've also talked about the what? The primitive node. So when you talk of the primitive streak, this is a very important thing that you need to take note of. For this, it's indicate one, the formation of the 3 germ layers, and also it leads to the commencement of the formation of the what? Of the nervous system. Though we have the main inducer, which is the what? The notochord, which plays a very big role when it comes to the formation of the nervous system. So one thing we need to understand is that by the end of the second week, where we had to observe the epiblast, okay, the epiblast and the hypoblast. So by the end of the second week, where we had the epiblast and the hypoblast, there is a groove midline depression. Okay, so there is this depression in the caudal end of the bilaminar embryology. And this groove, okay, this groove, it's more like something like a hole like or a line like. Okay, so this groove is just marks the appearance of the what? The primitive streak. a groove-like structure. So the groove-like structure which is existing in week 2, it's just leading to the formation of the what? Of the primitive streak. And then we need to understand that this primitive streak will normally start deepening. The primitive streak, which I've already shown on this diagram, will normally start deepening. And this deepening will normally start taking place during week what? Week 3. So the deepening will lead to the formation of the what? Of the formation of the primitive node, which is somewhere there. So when the primitive streak is formed, the next thing that will be formed is the primitive node. This is normally formed on the cephalic region of the what? On the cephalic region of the embryo. On the cephalic region of the embryo. If you can observe, there is a primitive node somewhere there, and this guy is the primitive streak. Okay, so let's quickly move on and look at what we need to understand. Then one thing we need to understand that the cells of the ectoderm, will start migrating toward the streak. Okay, the cells of the ectoderm will start migrating toward this streak. So remember these cells, okay? These are the cells of the ectoderm. So the cells of the ectoderm will start migrating this side toward the what? The streak. So when these cells of the ectoderm starts to move, remember there is a primitive streak here, okay, which is a groove-like structure. So these cells of the ectoderm, the already existing cells from the epiblast, they will start moving toward the what? The streak. So as they move toward the streak, they will normally lead to the formation of the third type of germ cell, and this third type of germ cell is known as the what? Intra-embryonic mesoderm. So the intra-embryonic mesoderm is as a result of the movement of the cells of the what? The cells of the ectoderm towards the what? The primitive streak. And this is taking place due to the fact that the cells in the ectoderm, they are consistently doing what? They are consistently dividing. Okay, so please make sure that you are able to understand. Remember, we have the endoderm there, we have the endoderm which is down there, we have the endoderm there, there. So these cells will start moving that side. As they move, they'll be moving toward the what? They'll be moving towards the primitive streak. Remember, there is a primitive streak which is a groove-like structure. So these cells, when they start moving like that, of the cells of the ectoderm, they will lead to the formation of another layer in between the ectoderm and the endoderm. So this layer is the one which is known as the what? The intra-embryonic what? Mesoderm. Okay, so this can be explained in terms of this diagram. Remember, we can talk about this as the endoderm layer, and then there is the ectoderm layer. These things have already been explained. And then these cells of the ectoderm will start moving toward the what? The groove, which is the primitive streak. The other name of the primitive node, it is just the Hense's node. So please make sure that you take note of that name. So these guys are just some mesoderm, those ectoderm. I think these I've already explained them on the other part. With this being said, let's quickly look at the formation of the main inducer of the nervous system. And this is the notochord. So the notochord is just a solid-like structure. Okay, and this solid-like structure, it comes from the what? The notochord plate. So the cells which are derived from the primitive node, remember, okay, the cells which are derived from the primitive node. So we have the primitive node here. So the cells from this primitive node, they'll start migrating what? Cranially. They'll start migrating cranially. So as they migrate cranially, they are moving toward the what? The procondyl plate or the procondyl membrane, which is just also known as the what? Bucopharyngeal membrane. So this bucopharyngeal membrane is the future mouth. This is normally known as the what? The future mouth. So my mouth, which I'm using right now, it is what? It originated from the bucopharyngeal membrane. So the cells from the primitive node, that deep structure, that node which was there, will start what? Migrating cranially toward the what? Bucopharyngeal membrane. Now, this result to the formation of what we are calling the what? The notochord plate. And this notochord plate will form a solid-like cylinder. And this solid-like cylinder is the one which we are calling the what? The notochord. Remember, the cells of the primitive node, and this primitive node which was created from the primitive plates, the primitive which was created from the primitive streak, this primitive node will produce these cells. These cells will start moving cranially toward the bucopharyngeal membrane. And then as they move, they will result into the appearance of the notochordal plate. And this notochordal plate will start folding itself into a solid-like cylinder. And this solid-like cylinder is the one which we are calling the what? Is the one which we are calling the notochord. So the notochord normally comes to underline the following. One, it comes to underline the future neural tube. And the future neural tube, this neural tube is the one which is leading to the formation of the brain and the what? And the spinal cord. So this brain and the spinal cord are leading to the formation of the central nervous system. So the central nervous system is normally comes from the what? The neural tube. Okay. So with that being formed, you need to understand that it also underlines the longitudinal axis for the what? For the embryo. With this being said, ladies and gentlemen, let's quickly look at the neuration, the main thing which we are interested in, the formation of the neural tube, the formation of the nervous system. And this formation of the nervous system, which is happening in week what? In week 4, it is called what? Neuration. So let's quickly look at what neuration is. So remember one thing that I've already stated. Neuration is the process by which the spinal cord and the brain are being what? Formed. Neuration is the process by which the spinal cord and the brain are formed, or this is the process by which the central nervous system is being what? Formed, because the brain and the spinal cord, they are forming what we are calling the what? The central nervous system. And this is taking place, okay, this is taking place in week 4. And then, yes, we are interested in knowing that the brain and the spinal cord are formed by the process of neuration in week 4. We need to understand that where is this brain coming from? Where is the spinal cord coming from? Okay, so with that, we need to go back and look at the 3 germ cells. Remember, I said there are 3 germ cells which we normally talk about. We have the ectoderm, and the what? Endoderm, which are the first ones to appear, the endoderm and ectoderm. Then we have the last one, which is the what? The mesoderm. of the three, the one which is playing a role in the formation of the brain and the spinal cord is the what? The ectoderm. So this ectoderm will give rise to a layer which is known as the what? The neuroectoderm. Remember that the ectoderm can differentiate in so many things, a lot of things, such as the upper layer of the skin, which is the epidermis. This ectoderm can go and make what we are calling the nucleus. But when we talk of the nervous system, the ectoderm will simply do what? Will simply change into this important type of what? This important type of layer, which is known as the neuroectoderm. So the word neuroectoderm, it is just divided into two. One, we have neuro part, which is talking about the what? Nervous system. So this neuroectoderm, it is the one which is leading to the formation of what we are talking about, which is the nervous system. And this nervous system is the thing where we are talking about what? The spinal cord and also the brain. All right. Let me quickly summarize this work using this diagram, and then I'll go on with more notes, and you are free to get it from anywhere. So the first thing is that the cells within the mesoderm, they are going to form the what? The notochord. Are you understanding? What you need to understand is that the notochord is being created, one, from the primitive streak, okay, and also this primitive streak is the one which is forming the what? The primitive node, and the cells of the primitive node will start migrating what? Cranially, okay, and when they migrate cranially, they will lead to the formation of the notochord what? Notochord plate. And this notochord plate will start being formed in a solid like cylinder, and this solid like cylinder, it's what we are calling the what? The what? The notochord. So the notochord can also be said to be derived from the what? From the mesoderm. It is created within the mesoderm, guys. This is the ectoderm. This is the ectoderm. Remember, this is the ectoderm, and then this is the endoderm, and then you have the what? The notochord within the mesoderm. Then what will happen is this notochord which has appeared here, it's going to weaken this portion. Are you getting that point? The notochord will start weakening this portion. So when it weakens this portion, this is a reason why it is believed that the notochord is an inducer of the formation of the neural tube, because it's going to weaken this. When it weakens this, look, it weakens this point, then the ectoderm layer will start moving in that way. Just like this diagram. I think this diagram I show. The notochord layer will start expanding like that, and there is a groove which is formed, and this groove is known as the what? The neural groove. Okay, so there we go. So there is a groove which is formed when the ectoderm start moving this side laterally, expanding laterally. Then there is a groove which is going to be formed, and this groove, it is the one which is known as the what? The neural groove. And then you have this side which is known as the what? Which is known as the what? The neural fold. Remember, we are interested in the formation of the nervous system, and for the nervous system to be created, there should be the formation of what? The neural tube. Okay, so in that case, this group has been formed, and there is the neurofold which have been formed. So the neurofold, the lateral neurofold will start moving this side and that side, midline, and they will cross at one point, they will meet. And when they meet, they will form a hole-like structure which is known as the what? The neurotube. Remember, the neurofold will start moving up, expanding, and then when they expand, they will start going midline, they will meet. When they meet, they will leave out a hole there. When they leave this hole is the one which is known as the what? The neurotube. Okay? Then, on the other hand, the mesoderm will lead to the formation of the what? Of the somite. The mesoderm will lead to the formation of the what? The somite, and then also the column. Okay, so we move on. All right, so let's quickly move on what is happening on day 18. So when you do 18, the embryo already consists of the 3 germ layers, which are now repeating the same thing: the endoderm, mesoderm, and the ectoderm. And then this thing, there will be a thickening of the ectoderm to lead to the formation of the neural plate. This thickening of the ectoderm, it's because of the presence of what appeared in the mesoderm, and that was the what? The notochord. Okay. With that being said, there is a formation of the neural groove, and this neural groove will normally appear on day 20. So the neural groove is expected to appear, or to begin to develop by what? By 20, because by 22 a neurotube has to be formed. By day 22 a neurotube has to be formed. Okay, so let's quickly look at the diagram. So this is just a diagram. By day 18 you have all the 3 germ layer. You have the ectoderm, mesoderm, and what? Endoderm. And then there is the formation of the what? The neural plate. This neural plate has to be formed by the what? By day 20. By day 20, the neural plate will start what? Will start forming. And this is taking place because there was something that was being noticed, and that thing was the what? The notochord. So this notochord, it led to the induction of the formation of the what? The neural groove. And that neural groove, it will turn into the what? The neural tube. And this will normally take place at the what? The 22. So by 22, like I've already said, by day 22, we need to expect the formation of the what? A neural tube. Then, a few days later, something will appear. A few days later, something will appear to give us a signal that yes, now the brain and the spinal cord are about to be what? To be created. So by day 22, the neural tube has been formed. Then a few days later, there will be something that will appear, remember, and that's something which will be the division, the 4 major divisions, which is the telecephalon, diencephalon, mesencephalon, and the rhodocephalon. But not all of these guys are going to divide. Some of these guys, they won't divide. So let's quickly look at how the picture of these guys will look like. So by day 22, there is what? The neural tube, which has already been formed at this region. Okay, then by 22, after some days, we need to expect the appearance of the telecephalon, diencephalon, mesencephalon, and the rhodocephalon. Take note of that. So let's quickly look at what happens next after these guys. So how does this happen? How does this happen? Okay, I've already explained this thing, but I'm now again trying to explain how this folding of the neural tube will take place. So remember what I said. There is a groove here. There is a what? A groove. This groove is known as what? The neural groove. So the cells on the neural fold, will start moving this side. Remember, there is a notochord down. There is a notochord down somewhere there. So these cells will start moving that side. Well, so when they start moving that side, they are leading to the formation of a hinge, which is known as the what? The median hinge, which is there. Then this side, because they are keeping on moving, they are keeping on growing. Even this side, they are keeping on growing. They will lead to the formation of the what? The hinge, the lateral hinge joint. So the medial hinge point which is formed, this is believed due to the signaling from the what? From the notochord, and it has the columnar cells. So you need to understand that the notochord, it's the one which has induced something, and this something, it has led to the formation of the what? The median hinge point, and this median hinge point is the one which will make more movement of these lateral sides toward the what? Toward the midline, of which this side, you know, we have the what? We have the neural faults. So the same thing will happen to the lateral fault, and this normally is believed is due to the presence of the nearby mesoderm. So when these guys move, this side and the other one moves this side, of which this is the neural folds which are moving this side, of which I've already explained this. You need to understand that now this will lead to the closure. So they will literally close there, and this will lead to the formation of the neural tube. So let's quickly move on and understand. So the closure which normally happens, by zipping, this normally happens, or this normally zips rostally and caudally. So let's quickly look more on what I have explained. Okay, so I've already explained everything. The only thing that I'm interested in is another word in which we need to understand that the folding and closure of the neural tube, it occurs first on the what? Cervical region. The closing and folding of the neural tube, it normally occurs on the cervical region. Then, due to the zipping, there are two pores which are formed. These pores have other names which I'm going to explain later on. There are two pores that are formed. There is the anterior pore and the posterior pore. The posterior pore, it normally closes by day 25, and the anterior pore is the one which starts. It's going to close by the what? Day 25, and 3 days later or 2 days later, which means by day 27 or 28, the other neural pore, it also has to close, and this happens on day 28. This simply means that the closure of both these guys is happening at week what? Week 4. So during week 4, that's where the anterior neural pore and the posterior neural pore are being what? Are being closed. Okay. I think, guys, when you talk of the neural pore formation, so these are just in other ways in which we can literally explain what normally happens. So at the end of the 3rd week of development, the ectoderm develops. And this ectoderm has the shape of the disc, and this disc, it has a broader region, which is the cephalic. So the cephalic is more broader than the what? The caudal region. So after knowing that, we need to understand with further development, there are an issue of the formation of the neural fold. There is an issue of formation of the neurophode, and this neurophode, they'll start moving medially. Okay, they'll start moving medially. So as they move medially, they are going to close to form the what? The neurotube. But before the neurotube is formed, there is a groove which is created, and this groove is created due to the induction which has been formed by, which has been caused by the what? By the notochord. And then this neurophode, since the embryo will look like that, so there is a groove. Okay, there is a neural groove, and there is a midline hinge. Then there are some more neurophodes this side. So the neurophodes will start moving in this other way, which is toward the midline, and at one point they are going to close, and they will lead to the formation of what? The neural tube, which I've already explained. So these are the same thing that I'm explaining in a different language. I'm just explaining in the what? In the different language. So the fusion, it normally occurs on the cervical region. The fusion will normally start from the cervical region, which is approximately on the fifth what? Somoid, and it proceeds in what? Cephalic caudal direction. That's why we said it normally takes place laterally or caudally. It normally takes place laterally or caudally, meaning when you talk caudally, that means that it's moving from the cephalic region going to the what? To the caudal region. Okay, so with that in mind, remember, when the closure takes place, we need to understand that there is a fusion, and this fusion leads to the formation of two important neuropores. The first neuropore we talked about is the anterior neuropore. The anterior neuropore is also known as the cranial neuropore. And the cranial neuropore, which is the anterior neuropore, it is the one which is going to close on day 25. The cranial neuropore, which is also known as the anterior neuropore, it is the one which is going to close on day what? Day 25. Then 2 to 3 days later, the caudal neuropore is also going to close, which means on day 27 or 28, the posterior neuropore or the caudal neuropore will close. And these two pores, they normally communicate with the overlying what? Amniotic cavity. Remember the amniotic cavity, which was formed within the what? This guy was formed within the what? The epiblast. This guy was formed within the epiblast, and this took place during week what? Week 2. Remember that. Okay. Then, with that being said, you need to understand that it is very, very important for us to understand what is required during the what? The formation of these guys. So what you need to understand that I think that has already been explained when the cranial neuropore closes, and also when the caudal neuropores closes. I've already explained that. So we need to understand that the closure of these two guys, the cranial neuropore and the caudal neuropore, it needs the presence of the what? The folic acid. This is the reason why during antenatal, mothers are given what? Folic acid. That folic acid is given to them just to make sure that the process of neuration can easily take place, to avoid some abnormalities. Okay, let's quickly move on. We are still on the neural tube formation. So you need to understand that the cephalic end of the neural tube shows 3 dilation, and the primary and the primary brain vesicles. So the first one, it is the prosencephalon, which is also known as the what? The forebrain. Then we have the what? The mesencephalon, and lastly we have the rhodocephalon. The prosencephalon will divide, and also the rhodocephalon will divide. Only the mesencephalon does not divide. This does not divide. This does not divide, and it normally take part in the formation of the brain stem, because sometimes the formation of the brain can be classified into two. So remember that the prosencephalon is capable of dividing. Rhodocephalon is also capable of dividing. That's why we had those guys whereby we talked about the telecephalon and also the diencephalon. So the prosencephalon, when it divides, it will lead to the formation of the what? It will lead to the formation of the telecephalon and the diencephalon. And then when you talk of the rhodocephalon, it will lead to the formation of the what? Of the metencephalon and also myelencephalon. So let's quickly move on. Now there are two fractures which you need to take note of. The first one is the cervical fracture, and this cervical fracture is just at the junction of the hindbrain and the spinal cord. So the fracture between the hindbrain and the spinal cord is the cervical fracture. And then the other one, it is the one which is found within the midbrain, and meaning it is within the what? The mesencephalon, and this is just known as what? The cephalic fracture. So the cephalic fracture is found within the what? The midbrain region. Okay. So when the embryo is 5 weeks old, so we need to understand that in week 3 in week 4, there was the closure of the neural pores, the anterior neural pore which closed on day 25, and also the posterior neural pore which closed on what? On day 27 or day 28. Then during the third week, during the fourth week, we need to understand that 3 things appear. We had the prosencephalon and the what? Mesencephalon and also rhodocephalon, and also 2 fractures. Now, when the embryo is 5 weeks old, meaning we are now in week 5. So during week 5, the prosencephalon will divide into 2, which is telecephalon and the diencephalon. So when you talk of the telecephalon, this guy is just formed by a mid portion and 2 lateral output getting called the what? The primitive cerebral hemisphere. So the telecephalon is just formed by the what? The mid portion and the 2 lateral outpocketing, which are known as the what? The primitive cerebral hemisphere. During the same week, which is week 5, the prosencephalon has divided into 2, telecephalon and also what? Diencephalon. And this diencephalon is normally characterized by outgrowth of the optic vesicles. Okay, let's move on. Now, we need to understand that in week 4, we had the mesencephalon and rhodocephalon. Now, there is a deep furrow which exists between the mesencephalon and the rhodocephalon. And this deep furrow is known as what? The rhodocephalon, the rhodocephalic isthmus. So this is just a furrow which exists which exists between the mesocephalon and the rhobocephalon. But within the same week 5, the rhobocephalon divide into two, which is the metacephalon and myelencephalon. The myelencephalon is the one which is normally known as the medulla oblongata. So myelencephalon is coming from the rhobocephalon. Then the metacephalon is also coming from the rhobocephalon, and this metacephalon is leading to the formation of the pons and cerebrum. Pons and the cerebrum. Take note of that. Then remember, there is the mesocephalon which doesn't divide. So when we talk of the brainstem, we need to understand that when the brain we divide it into two, we need to understand that there is these three guys which makes up the brainstem. So you have the mesocephalon, metacephalon, and the myelencephalon. These three, okay, they form the brain stem. And this takes place in a case where the brain is being divided into two. Okay. Now, there's something that we all need to pay attention to, and we need to understand that the lumen of the spinal cord, the central canal, is continuous with the brain of the with the brainy vesicles, such that we have the cavities of rhobocephalon is just the fourth ventricle. So the cavity of the rhobocephalon is just the fourth ventricle. Then the cavity of the diencephalon, which is coming from the proencephalon, this is just coming as on the third cavity. Then we have the cerebral hemisphere, which are just the lateral vesicles. Take note of that. Please take note of what we are just simply discussing. Then, with that being said, we need to understand that the lumen of the mesocephalon, which did not divide, it normally connects the third and fourth ventricles. The lumen of the mesocephalon, because the mesocephalon is between the proencephalon and the rhobocephalon. And then we know that the proencephalon will give rise to the telecephalon and the diencephalon. So the lumen between the diencephalon and the rhobocephalon, that part is connected by the lumen of the mesocephalon. Okay, so that's one thing we need to take note of, whereby I've said the lumen of the mesocephalon is connected third and fourth ventricles. Now, this lumen, it normally becomes very narrow and is then known as the aqueduct of Sylvius. So the lumen which is taking place due on the lumen of the mesocephalon, which is taking place between the diencephalon and the rhobocephalon, this lumen, it becomes so narrow because it's connecting the third and fourth ventricle. It becomes so narrow and it leads to the formation of what we are calling an aqueduct of Sylvius. Okay, let's quickly move on. Then we need to know that each ventricle communicates with the third ventricle. Each lateral ventricle communicate with the third ventricle through the what? The intraventricular foramen of Monro. Yeah. Now, let's quickly move on our last part, which is just the what? The brainstem. So sometimes the brain is divided into the brainstem and the what? The higher centers. The brainstem is made up of the following. One, it is made up of the mesencephalon, which is the midbrain. Two, it is made up of the pons from the what? Metacephalon. Three, it is made up of the myelencephalon, which is the medulla oblongata. Then we have the higher center. Remember, the brainstem, when the brain is divided into two, it will give rise to the what? It can be divided into the brainstem and the what? And the higher center. So the brainstem alone, it's made up of the pons of the what? Metacephalon. Then we have the midbrain, which is mesencephalon, and also the medulla oblongata of the myelencephalon. Then when we talk of the higher center, this just consists of cerebrum, which was part of the metacephalon, and the cerebral hemispheres. So when you talk of this brainstem, the brainstem is a direct continuation of the spinal cord, and this guy has the similar organization. So guys, please make sure that you go through this video, you go through the notes, and make sure that you go through exam questions. In the meantime, this was Dr. Possibility from Excel Academy. Thank you very much for joining me. Have a blessed day.